nutraceuticals, and we're here to share a dose of knowledge on what works, what won't. We'll entertain you and we'll show us innovative.
Hey everybody, welcome to another edition of The Dose. I'm Todd Runstead from Next USA,
I'm Devon Golom from Step Change Innovations, and
and
we
we're the,
are
the Nutri-
Nutraceuticals.
God.
We're
We're
not,
recording this first thing in the morning. Clearly, the caffeine has not kicked in. Here's to
yeah.
alkaloids, everybody. The world's most popular drug is actually caffeine. It's the alcohol from that wonderful uh, 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 extract of a dark bean called the coffee, right? And I think life has really improved in at least the functional beverage world once caffeine went out of the coffee cup. I know, as for myself, I never liked the taste or the smell of coffee. The only thing I really like about coffee is the vibe of coffee shops. Um, and so now you can get it in shots and sodas in all kinds of stuff. You can buy a kilo of powder and try to overdose if you want. Alkaloids. It's the caffeine, cocaine. All those things that really kind of get you going. And
Oh,
obviously, I'm going, and Devin, I know you are too. I can tell by your right eyelash, eyebrow. See,
yeah.
it's
My
not quite
eyes are
kicking in.
not even today. I'm not quite sure. I'm just wondering how you're going to connect all this into introducing our guest for today.
Let us introduce our guest today, ladies and gentlemen. We're hoping that Blake Ebersole, old friend, he has been writing for Supply Side Supplement Journal. He is president and founder of NaturPro Scientific, and he is really expert at getting lively, deep conversations going on LinkedIn.
Oh, yeah.
So he's a live wire, kids. So if the caffeine has not kicked in, sit back, hold on. Ladies and gentlemen, Blake Ebersole, tell us something that we don't know about you. Good morning.
Well, first of all, I started drinking coffee out of borosilicate a while ago because, everybody pays attention to their coffee ritual in the morning, but they're not thinking about, well, what is the composition of the glaze that's on your coffee mug? And so, non-leaching, and then, it's got the graduation there, and so you know exactly how much you're drinking. I got reverse osmosis water and all those things, and so. Yeah, I've kind of tuned that into an art. I always thought it was interesting the reason why the Industrial Revolution and human advancement came along. I've heard is that particularly in Europe they switched from drinking beer and alcohol in the morning to coffee, and so suddenly people who are working a half day, maybe not so well, that coffee made them work those 10-hour days. So hey, we can credit coffee and caffeine maybe with where we're at right now, which is I guess half good and half bad. But hey, the reason why I really want to be on this show is because it's proof that scientists have more fun, and we're
That's
funnier
right.
too. It's scientifically proven, although scientists probably publish those results, that we are funnier. But we have to have a sense of humor when we see people around the world, that don't understand the world like we do. And so, really thanks for having me on. And I've
Yeah.
focused so much on, supplements, natural products over the past 20 years and, the technical side, but, I've also been interested in, how do we share and how do we educate, people on the real deal. And, obviously right now there's lots of misinformation and people that just don't understand because maybe you just don't have the experience or you're getting your information from social media. And so there's a role for us scientists here to kind of try to set the record straight sometimes. And I'm fortunate to be in a position where I can say what I want as owner of the company. I don't have to get approval from the boss. And so yeah, when a thought pops in my head, and it seems to be a little bit different than what other people are saying. I'm just going to quick write it and post it for the world to see.
No, I
Yeah.
love that you do that, though. You're one of my favorite follows on LinkedIn because you're putting the truth out there and you're unabashed in how you talk about science and how you talk about the effect of current events on, what's going on in our industry. So thank you for that. You give me, yes, you give me a lot of courage.
Thank
Thank
you.
you.
So, should we be concerned about science in two ways? And let's jump down this rabbit hole. One is, this was like, I remember 15 years ago, something came out about how abstracts, so many people, they just go to PubMed, you look at the abstracts and you say you read a study, and abstracts are not entirely accurate so much of the time. A. B, AI. So we've already had a problem with studies being pulled after the fact, because, for whatever reason, and we can dig into that. But also, there's this whole issue with AI generating a study on its own, and it's all bullshit. So, what's the challenge for scientists and education in an era of shadows?
Yeah, it's such a intractable appearing problem, but it's important to remember that while science might be perfect, it is a process. Number one, and
Yes.
number two, there's humans involved. So humans are administering the science, they're understanding, they're communicating the science, and so we have to think about human incentives, human motives, for example, to publish information. And particularly when your paycheck is reliant on the publication, well, you're not going to spend 2 years on a study that has bad or no results and then not come out with a publication because that's going to look bad. And so it's publish or perish in many cases. And so you're going to keep working until you find some data that looks good, and then you're going to tout it. You're going to really make it try to look good. And obviously the abstract is just a snippet or a cross section of what's actually in the paper. And scientists understand that people only look at the abstract, oftentimes because the abstract is the only thing that's free, and then you have to pay $35 to read the full text. And so,
Right.
Even when you get into the methods of studies, like only people within your discipline know whether the methods you used were good or not. And so that's a big part of it. And then the other part too is science has evolved, and so when we look at papers from 20 or 30 years ago, especially in nutrition, the methods they used were maybe not the best, but it was the best they had at the time. And so that's why when we say, oh, that's an old paper, well, it's not just that it was rickety and it has bad joints. It's that there's better methods that came out since then, and sometimes we might have conflicting evidence because the old method wasn't as precise. The newer methods are better. And so, unless you're a scientist and you follow this stuff and you're involved in the publication process, you're just looking from the outside. And so, it's easy to blame, big pharma shills and all this type of stuff for inconsistencies that you see. But until you've actually been part of the process, you don't really know. And it's a severe Dunning-Kruger syndrome effect where the less you know, the more you think you know. But yeah,
Right.
we're at the point where we see all these opportunities and all these missing pieces at this point. Even in nutrition, the methods aren't perfect, and we're bound by practical things like the budget of the study and the labor available to perform the study, and so that we do the best we can, but the results sometimes aren't the best. And so when we say scientific consensus, well, that means, okay, independent papers are agreeing with each other. It's not different people agreeing with each other. Consensus is, okay, somebody from around the world did a similar paper or study that you didn't even know about, and hopefully maybe their results agree with yours without knowing what you did. But sometimes they're different. Well, why are they different? Sometimes it's methods. Sometimes we just like cherry pick the data and try to do the best we could because we're scientists, and our paycheck is closely tied with what we publish.
Yeah, you hear that a lot of in the mainstream press, how so much nutrition science research is performed by brands, which really means mostly ingredient suppliers, and maybe it's universities that are doing it, and then it gets taken up. But it's always it's a trick because there's a tendency to compare nutrition and nutrition science research with pharma research. But they're different because no one is deficient in a statin or semaglutide, right? It's just this is a single chemical constituent molecule, a pharmaceutical that you take in, you don't have any in your body, and it has this profound effect. Whereas with a nutrient, you know, if you're talking with an Italian person who eats a lot of, marinara sauce, and then you're going to do a study with lycopene, well, there's already a ton of pre-existing lycopene in that person's body, and so when you supplement with 100 milligrams of it, maybe you don't see that much of an effect. You would see a bigger effect on someone, who doesn't have, excuse me, a lot in them already. And so I see this all the time, and I think it's changing a little, but it's something that I've been railing against for years, is you need to have baseline biomarkers at the start of every study so that, okay, this is the state of affairs where it begins with, and maybe you divide it out into quintiles, and then you go ahead with it, and then you could see the difference in different quintiles, and then that gives you a different conclusion that you might draw from the study.
It's fun being in between you guys because we've got Todd here who wants like the ideal study, right? Look at all those base markers, have lots of people so you can divide them into your different quartiles, quintiles, whatever. Yeah. And then you've got Blake over here who understands the reality of budget constraints, of time constraints, of having to get data, get something out there that you can publish. So, and one of the questions that was going through my head listening to both of you, and I'm going to pose this to Blake, like, if you were to, like, what do you look for when you're designing a study? Like, what would your ideal study include? And we can pick some fictitious ingredient to look at. Like, what would your ideal study be in terms of what you're measuring, population size, duration, etc.? And what are the hurdles that you come up against most often when you are planning those trials? And where do you reach a happy medium?
Yeah, that's an excellent question. And going back, starting with Todd's earlier statement is, the difference between pharma and nutrition studies is, pharma studies are the easiest freaking thing you can do because you're looking at one individual molecule of which the population isn't taking anything else from food, and then you're looking at one outcome that through the pathway that one compound works. Whereas nutrition studies are some of the hardest studies ever to do. Because there's so many confounding factors. And you say, okay, well, if you're studying the effect of a nutrient, you can't tell these people to remove that nutrient from their diet because then that would be unethical, right? Because you need a certain amount of that nutrient every day. And so, we've worked with, CROs, contract research organizations who have excellent expertise in pharma, but when they get to nutrition and nutraceutical studies, they're a little bit lost because they don't really understand these nuances or intricacies. And it's also worth understanding that each of us is an individual. Each of us has our own biochemistry.
Oh, yeah.
So it's very difficult to generalize studies, even on, even a phase 3 study, 150 people. Well, out of those 150 people, what was the spread, what we call inter-individual variability among their response to the intervention? And how does that result from the study represent me as an individual, how I'm going to respond? And then you get into like, okay, yesterday I felt stressed, today I don't. So even within an individual, depending on the time or day you take the measurement, well, so yeah. So, I posted the other day on cortisol, like we all have cortisol awakening response. It spikes in the morning. Well, some of us spike more than others because we're more stressed or just because of our biochemistry. And so, in the old days, 10, 20 years ago, there was no way to really try to control for these factors or select the patient population because we just didn't have that, data on the people coming in the study. But now with big data and, these long questionnaires that people are answering, well, you can pick the group that most represents, who is going to respond in the study or in other cases represent the largest diversity of group. For example, picking people of different ethnic backgrounds and things. But to your question, Devin, I think you really have to understand and go back through scientific method. What are the answers what are the outcomes or the answers you're trying to answer the questions for, and what are those questions? And so a lot of nutrition and supplement companies are doing this studies just based on what everybody else has done, but they're not really thinking about, okay, number one, what are the current methods? How can we do this differently to get more consistent results? And then second, how do we show the difference of our product or ingredient versus everybody else that's done the same thing? And so you say, okay, joint health, for example. Well, historically studies have been done over the course of 60 or 90 days. But what consumer do you know that wants to wait 60 or 90 days until their joints feel better? And
Right.
so
I
you
mean,
get into study
glucosamine
designs.
was 6 to 8 weeks. That's like the
That's
end of
right.
the
And
second
so
bottle.
so yeah, you say, okay, well, it might be a gamble, but let's see if we can get an effect sooner than that. And so that's where, within joint health, because everybody knows Tylenol or ibuprofen, you get a response in hours. So why are you going to take, turmeric or whatever? If it's going to take 3 months for a response. And so those results need to be applicable. Otherwise, you're just saying supports joint health, and then you're saying the same thing as 100 other people, and you're not positioning yourself in any way with the study.
It's
Yeah,
so funny you're talking about joint health because it just so happens I have two papers here on joint health on an ingredient called TamaFlex. I'm going to do some shameless promotion for my co-host here.
that's
But
right.
one of the to your point, one of the papers was 90 days and another one was 30. And not everything not you don't I mean it's apples and oranges with drugs and supplements sometimes because drugs quite often give an effect right away and a lot of supplements don't. So but if you can document a clinical impact within 30 days, then you can basically tell your consumer, hey, take this, do the whole bottle before you decide whether or not this works for you.
Yeah, and that's worked out with the Tamiflex, where we have results that are starting in only 5 days. And so we're getting supplement brands that are buying this ingredient, and they're putting that 5 day claim right on the front of the bottle, and that's
The
powerful.
makers of Tylenol, as it just so happens.
Right. Yeah. I mean, Tylenol got into, jumped off of OTC and into supplements, and they're using Tamiflex because they love our 5 day claim. And so the interesting thing about just the joint health category in general is it's flat overall, but glucosamine and chondroitin are collapsing. And so that gap is being filled in with these faster acting things. That's what consumers are like, really? I got to wait till the second bottle and maybe I'll get it, a result. Whereas, okay, well, if I'm going to start to feel something, after a week, I'm all in. And I just want to point out one of the things that we do at Next in our study design is what we call, and sorry for everyone who think I'm going to have a commercial here, but I think this is important as far as like the bigger issue on how we design clinical trials, what we call the 4 pillars of validation. So you have the patient responses, it's like, oh, I can feel a difference. And this is, there are CROs that will, give subjects an iPad and it's like, okay, we're going to remind you every day to take this thing and then tell us how you feel. And that's great, as far as that goes. But that's only, to us, that's only just one of the pillars. Because then you need a physician assessment, expert eyes confirming, visible health improvements. Then you have biomarker analysis. And that's what I was talking about before, is like, you should start with that just to see, okay, where are you at on the baseline? Because then if those things move, like CRP or IL-6 or some of those inflammatory markers, it's like, okay, well, that doesn't lie. That's objective, versus a subjective patient response. And then finally, there's the physical measurements, where it's like a strength test, a 6 minute walk test, flexion, how does it move? And so combined, where you have all those things, it's like, yeah, like someone is saying it's good, an expert saying, yeah, that looks good. The biomarkers are like, yeah, something's happening here. And then you're actually, wow, I could move faster, I could go further. You combine all that, and that is what sells supplements. You know,
Yeah,
that's
and what you mentioned, Todd, is so important because now we're getting into an age where CROs are offering these virtual studies, and much of it is based on subjective experience. But they're not going to the doctor to get a radiological exam. They're not doing so. So the ability for bias to creep into these studies
right.
and the limited nature of actually measuring whatever blood or biomarkers kind of limits the power or the confidence you have in those results, right? And so, that's why I've always thought, well, virtual studies might be great or everything, but they're kind of missing a few things that we would want in these types of studies.
Yeah, I
Um,
mean, it's a good start, because it'll say, okay, like it's a cheap way of saying, okay, we seem to see something, but then I
well,
think you got to like invest and
Right.
I'm glad that you brought that up because that was one of the next questions I wanted to ask. Because Todd and I have touched on this in a prior episode. I'm very bullish on decentralized trials, and maybe some of that is because I've read papers about how women are excluded from a lot of trials, and one of the barriers is that we're often the caregivers and we're the ones responsible for the kids, and we're the sandwich generation, so we're taking care of our parents too, and it's harder for us to get to some sort of center to do the trial. So being able to do things in the comfort of our own home can help with that a little bit, and can also help with your with the geography. You can get people all over the country if you're doing one here in the States as opposed to Cleveland or Chicago or Denver or something like that. But I also understand that there's a lot of things that we measure where sometimes questionnaires are the gold standard, like the PSQI, the Pittsburgh Sleep Quality Index for sleep. But we also now we have wearables and actimeters that can figure out, are we moving a lot when we're sleeping? Are we restless? Things like that. So I'd just be curious your thoughts on like where decentralized trials can shine versus where you really have to have that doctor assisted clinical evaluation or whatever. And then I'd also be curious if you see that changing, because we do see a lot more technology coming out where someone can get blood samples at home and send them in, as opposed to having to go in for a blood draw. So what's the state of it now? How will
people are
it
drawing
change
their own
with
blood.
technology? Yeah, so there's a company, I believe it's Strawberry Health, if I'm remembering right, that is about testing your hormones more frequently, because
Now, is that just a pinprick, or are they like sucking it out with a
it's this
syringe?
thing that you stick it in your arm, and it kind of like it takes a little bit of blood over time. I think it's like 20 or 30 minutes or something, but like it's more of a sample than a pinprick, or just giving because you can send in like a bleed on like a little piece of gauze or whatever, basically, and send that in to get like lipid testing and things like that. But they've designed this thing that it's kind of like a CGM, where you just stick it in your arm and then collect the blood and send it in. And that's kind of interesting. So if we have more standardized devices like that, where we can actually get the biological samples to send in, I mean, and we're already there with poop. So, I mean,
True.
with virtual colonoscopies and things like that. So, will that change the game for decentralized trials in the future?
Yeah. For those who
Yeah,
are squeamish about taking their own blood, I give you the poop test. If you can,
that's right. No, absolutely. So you mentioned a lot of great, advances in the ability to do these centralized trials, and certain categories like sleep. Well, the standard isn't to make you go in and do go to sleep in the doctor's office. It is, the clinically validated tools are these subjective measures.
Yep.
And so, yeah, certain categories of outcomes, I think you can say that. Obviously, anything doing with gut health, you can send a bag of poop in and get it cultured.
Just
Um,
ship me to
but,
the
but
lab.
but on the other side, like, we should caution against the zealousness of business interests to claim that you can do all these things with a small amount of blood, for example. And if you remember, that's what Elizabeth Holmes went to prison for, is because she was claiming you could do 300 biomarkers, with Theranos. And so, as a chemist, as an analytical chemist background, I know, I understand, like, okay, you need a certain amount of blood, and then it needs to be spun off if you're going to get a reliable. But yeah, certainly, like, because of the diabetes and everything, like that whole area has been pretty well validated because it's run at scale. And so you say anything, CGM or blood prick, but also caution against people who are trying to do shortcuts and it's not really scientifically validated. And so yeah, there's always people that are going to say, hey, we can test this stuff, but when you actually look at it, you say, hey, where's your validation? And they're like, well, what's a validation? So yeah, there's journals that publish validations on this stuff, and until you publish your validation, well, it might as well just be Elizabeth Holmes and Theranos.
No, fair
So
enough. I mean,
what
it's
do,
just oh, go ahead, Todd.
what do you think's more valuable? And this is maybe an unfair question. One of these sort of subjective patient questionnaire trials that you might get with an iPad, or a rat study.
Yeah, certainly, because those are clinically validated. The rating scales are numerical. You can do numerical stats on them, and yeah, they're totally viable. But yeah, that's certainly doing that in humans is better than any kind of animal study.
I would agree.
All
But
right.
yeah, but in terms of advances, like, so, okay, historically, for example, iron. So iron is one of the most deficient nutrients in the world. More than 1 billion people are deficient, and then a large percent of people are iron deficient anemic, which is you know chronic fatigue, and basically they can't live a normal life because of their problem. And on the flip side, that when they take the iron supplements, they're getting iron toxicity from all this iron, and it's not well absorbed. And so, but a lot of the old iron studies were done simply on iron status, but that doesn't give you the full picture. There's
And probably
actually,
on men too, right? And there's a
yeah,
big
well,
difference in.
if women are 15-20% of the population of women are iron deficient, still 5 to 7% of men are, right? And so yeah, and children as well. Unless your kid is eating a perfect diet, you know, they're probably.
Yeah.
And you think about the daily value of 18 milligrams. Well, a quarter pound hamburger only gives you, say, 3 or 4 of those milligrams. So you say, well, okay, if you're gonna buy a $60 porterhouse, well, that's gonna get you there. But you look at the
At what
shortfall
cost?
of the typical diet, iron content of diet versus daily value, and in athletes and menstruating women, you actually need more than 18 milligrams because you're losing that, you're turning it over. Um,
So
but yeah,
there's
go ahead.
Oh, I was just going to say there's two questions that I want to ask from this. And one, I do want to talk about gender dynamics and how that plays into variability in clinical trials and things like that. But going to the iron specifically, so a lot of people think like, oh, iron, you just test hemoglobin levels. But we had talked offline a little bit about how it's much more than that. Can you expand a little bit about that and talk about how, going after other biomarkers than what seems the most obvious tells a better story about how the ingredient is working?
That's right. And so, you think about the body in different compartments, right? So you have your plasma compartment, you have your RBC compartment, which is the part that's carrying the hemoglobin and the
Yep.
oxygen. And
Red
then you
blood
have
cells for the uninitiated out there.
yeah, and then you have storage iron, which is in your bones and is gradually released. And so you say, okay, well, hemoglobin might be great, but it's only one compartment of your whole body, and it's only a snapshot in time. And so, let's say you had more than normal iron over the previous week. Well, your hemoglobin might skew high. If you haven't been getting enough iron the past couple of weeks, well, it's going to skew low. And so, the other part about hemoglobin is normal range is only 12 to 14 units. And so, like, well, what is a 12.5 versus a 12.9? Does that mean you're increasing? Well, so then, so you get in all these like questions like, okay, well, of what meaningfulness is this one marker when you don't know your other markers? And so, looking at iron status or hematocrit, looking at the full picture. So ferritin is really what a lot of people are focused on, especially in terms of iron deficiency and anemia, because that's the storage protein. That's how you
Okay.
know whether you're over time. It's kind of like comparing fasting blood sugar to HbA1c. A1C is this, marker that represents over time
Yep.
versus fasting blood sugar is just a
Yep.
snapshot. And so now that these tests have, the panel has become more cost effective and cheaper, as it were. It gives you this ability to get a bigger picture. And if you follow like Iron Warriors group on Facebook, you see these people going through and trying to figure out their results and trying to tie that with what they're taking and why they feel so bad or why they're feeling better. And there's still so much to learn and understand about, what's the right iron supplement for them, because there's so much marketing out there of, oh yeah, this is better than that and all that type of stuff. Well, it doesn't matter until the rubber hits the road and you have these decent clinical trials. So now we're finally get to the cost-effective point where we can do this stuff. Historically, this stuff costs $2,000 or $3,000 to do a full panel. So it wasn't, nobody wanted to spend $3 million on a study, on a pilot study, because that's how much it would cost. Now it's more cost-effective, and so we can get better data because it's just a lot less expensive than it used to be.
And the ferritin, that's just a blood test, right? It's not like you need to get a bone biopsy or something like that to.
That's right. It all
Okay.
comes from
Cool.
blood, and so
Nice.
then you say, okay, well, how does blood plasma ferritin represent bone ferritin? Like, oh yeah, well, that's kind of interesting too. But ultimately, the rubber hits the road when people who are iron deficient and feel the pain, the poor quality of life, the fatigue, all that stuff, and they're starting to feel better, and their markers increase, and then whatever they're taking, well, it's not giving them a stomach ache. It's not, and so they feel it. And so, like Todd said, you want to have the markers, but what does that mean? Hopefully, it means better quality of life for the people who are taking it.
Now, I've been on an iron supplement for several years, and I figured out it wasn't necessarily that I was deficient in iron, but my platelets were always jacked up, and they did put me on some iron stuff for a while, and figured out that was why they went up, because I was deficient in iron. And I have to say, after like being on that supplement for about a month, I did notice a huge difference in my energy.
Interesting.
And still take it to this day.
Let's dig in a little more into the whole iron story. Are there timed release iron? Because would that help with a person's like energy levels for someone who is deficient in iron?
Yeah, so iron is like a riddle inside a conundrum inside an enigma, and because it's part of the Earth's crust, and the core of the Earth is made mostly of iron, and so obviously as humans evolved and animals evolved, we required small amounts of all of these elements, right? But iron, if you look on a periodic table and and you've seen an iron frying pan turn into rust, it's highly reactive and oxidizes. And so the body has, our biochemistry has evolved to very tightly control the amount that is getting into our bloodstream and getting stored in all these things, because too much is toxic, right?
Hemochromatosis.
So, we've been raised under this paradigm of, 65 milligrams of iron in a supplement, when in fact our body can only absorb 1 to 2 milligrams per day. So that's a result of the iron supplements we're taking not being very bioavailable. And so if you go back, millions of years ago, or at least, when humans started to be, hunters and foragers, they're getting a lot of their iron from meat, right? And so the form of iron in meat is bound within the heme molecule, which is porphyrin complex. And it's when you consume meat, if iron from meat, it's the iron is protected by the heme molecule, and it doesn't induce that toxicity, right? But then in the 1900s, when we started to develop iron sulfate and some of these things that were mined from the ground, they increased iron status in individuals who were sorely deficient, but it also causes toxicity because it's not in this kind of protein bound form. And so the literature over the past 20, 30 years has differentiated between non-heme, which is coming from plants or coming from iron sulfate or mine sources, versus heme. And so they find that heme iron is between 2 to 4 times more bioavailable, but it also doesn't induce this toxicity. And so less is more. So only 3 to 6 milligrams of iron is increasing iron status versus studies on non-heme iron are using 65 milligrams or more.
So
Wow.
interesting.
That's a
But
huge
it's
difference.
very difficult to isolate heme from meat because it's very energy intensive, and you got to do all this extraction and stuff, and so that's why it hasn't been really commercially available. And how do you extract it intact without it breaking down? So we had heme iron polypeptide, which is a fragment of the heme, but it's unpredictable. Like we really don't even know the molecular the molar percent of iron in heme iron polypeptide because it's not predictable. And so a company in Norway who's been fishing salmon for more than 100 years
Leroy
figured
Seafood,
out,
right?
So
Yep.
the blood of fish is a big waste stream, and when they're processing seafood, and they wanted to figure out what to do with it. And so they looked at all the studies and said, hey, this could be a good source of iron, but we have to preserve it intact. It can't be degraded. It can't be like a polypeptide form. And so it took them more than 10 years to figure out the process. Well, finally got it into clinicals, and they figured out that at 6 milligrams of iron, you're doing things that are better than what the 65 milligrams of iron sulfate or whatever are doing, and it's increasing ferritin, which you don't find with conventional forms of iron.
So interesting.
And
Upcycled salmon, is that what we're talking here?
Yeah.
That's
That's
right.
so cool.
And so Norway and Scandinavia are very focused on sustainability and be very involved in using 100% of the fish. And so that includes the scales and the everything like that. So there's some parts of the fish that are being used for biotech as like reagents for fibrinogen related reagents. And it's really cool how they're able to it takes a lot of time, it takes a lot of money, investment, but you can take these certain parts from a living fish that used to be waste product or fed to farm animals and create very high value ingredients from them. But yeah, you have to be invested in it. You can't just be greenwashing or greenwishing and saying, hey, this is upcycled. Well, what does that mean? Like, is it intentional? And what does it mean for the benefit of the consumer? Because it doesn't matter if something is green or sustainable if it doesn't really provide a sustainable benefit for the consumer.
And then you were saying that you could use a smaller amount of this salmon derived heme iron compared to like your commodity mined iron, right? So I'm sure there's a cost to that, but then you would want to communicate the benefit to that, right? Because there is that problem with too much iron, and I mean, is that less of a problem with women because they bleed it out once a month?
Yeah,
Is there a
well,
technical
so
term?
the product is called SalmoFer because it's salmon, and then fer is iron.
Right.
But yeah, it was when we planned the study, it was a little bit of a gamble to say, okay, will 6 milligrams of iron be enough to move the needle for people? And because most studies use much higher doses, but as it turned out, the results showed that it was enough. And there were some older studies showing, okay, very small amount of heme iron, or altering your dietary status with more servings of meat that provides a little more iron does tip the needle for people. But we were surprised to see how quickly it happened. So we did a single day acute study where we did PK and found that, wow, it's actually doing something on the first day. And then within 2 months, we're substantially increasing ferritin or storage protein from the deficient range into the normal range. And obviously we're the study I'm referring to, it's not yet published yet. It's about to be submitted, but the study was very interesting because we included women, menstruating women, two groups. One who were just considered deficient, which is not yet anemic,
Yeah.
and then also a separate cohort which were, fully anemic. And we wanted to see, okay, does the same dose or same program improve it more for people who are deficient? And so, looking at the raw data and the stats, we do find that people who are more deficient benefited more, which obviously you would expect, right? And
Right.
so, but it's kind of, magical in the sense that the same dose can help different
groups.
people of different status in a way that benefits both of them without the toxicity or without any of the
That's so cool.
negative side effects of typical iron.
So not to bring the conversation back to poop on purpose, but it is a common side effect of a lot of iron supplements that you have constipation issues. Does the bioavailability in the heme version of iron, does that tend to not induce constipation as much? Is there any correlation between the type of iron and GI issues?
Yeah, that's a good question. So what we found in the study, and we were expecting, maybe a couple people out of I think there was 45 in the study. We were expecting, okay, a couple people report something. Well, so we were surprised to find that none of them reported any issues.
Oh, good.
Which, I mean, not to say that's generalizable to the entire population of people that are going to take it, but the amount, the relatively lower amount of iron, as well as the fact that it's bound, within this complex, it's very similar in form to what you would get from a couple servings of meat.
Yep.
And so if you say, okay, if you don't get constipation from eating a couple servings of meat, well, that explains why we don't see these side effects. And the toxicity of iron goes beyond just the constipation issue, because it's killing off the good probiotics in your gut, and then it's allowing pathogens to grow, because there's certain pathogens like E. coli that love this iron-rich environment. And so you say, okay, if you're and then because it's highly oxidized in the gut, well, it's tearing up your intestinal lining, and so like, if you care about your gut, and then and that's why, like, in iron, it's like they've gone from giving people tablets to making them come in every 2 weeks for an injection. It's like it's 2025. There's no reason why we should make people have to go in and get an iron injection. We should have better solutions for improving your iron status.
Agreed. Agreed. Can we go back to clinical trials for a minute?
Yeah.
So we touched on women and gender dynamics and things like that. So one of the reasons that women have historically been excluded from clinical trials is also because, surprise, our hormones add a lot of variability. I was trying not to laugh when you were talking about inter-individual variability. Did I get that right? Because we're different from day to day, especially when we're in our normal menses years. So I've always thought that if you're doing a study on women, just have more women, because then you have more data and you can separate them out by, age, like what, regular menstruation years, perimenopause, menopause, etc. Would you agree with that? Like, how would you design a clinical trial with women to help overcome some of that hormonal variability?
Yeah, that's great. And I mean, it makes me remember a time going back a few years ago. I was involved in a study, and the study had about 2/3 men and 1/3 women. And which is how you see a lot of the ones that are like, yeah, it's in 40 participants. Well, 2/3 are men. In some cases, there are more women for whatever reason, if it's, university-based study or whatever, they're getting students, So, so there was a difference. There was a change in one of the aspects that might have been hormonal related or may not have been. And so the question was, well, because we had so many males in the study, did the results still apply to women? And so sorry about that. The answer.
That's a great ring.
That gets me fired up for calls. Yeah, the
Yes.
Rocky theme. No, but the answer was that unless the intervention acted in a way that would be expected to be different in men than women, then we shouldn't anticipate that it makes a difference whether you're man or women. Of course, that's one expert's opinion, and then you say, well, the flip side is also that, well, women are more complex than just having different hormone levels, and so. But yeah, you have to be thoughtful about it, and it's not necessarily true that every study would be different if there were more women in the study, but you also have to think about the nature of the intervention, mechanism of action, all those things. And I agree that women have been underrepresented. Different ethnic groups that maybe
Oh yeah.
absorb nutrients differently might be underrepresented. And so yeah, it really matters who's in your study, and not just that they're meeting the inclusion exclusion criteria, because I've almost never seen a study with the inclusion criteria being it must be about equal men or women, right? It's all this other things. Okay, no medical, but hey, why can't we make sure that it's roughly 50-50? It's just a little more extra work, but so clinicians are always thinking about money and time and all this stuff. But you say, okay, it's the sponsors, it's us who are directing the study design to the CROs, and we need to make them think about, okay, what should the clinical study design be beyond what is historically used and status quo.
Do you think you wouldn't really have that male female sort of issue in pharma studies because it's such a powerful compound that you're introducing to a body that it would sort of overwhelm any hormonal variability that you might have between genders? What do you think about that?
Yeah, again, it depends on what the outcome of the study is, but if there's anything related to hormone, you say, but, men and women are more similar than we are different, right? And so obviously there's small biochemical differences, but you don't want to prefer, like, if a drug is intended for a certain population, it's intended for both genders, unless it's prostate cancer or something like that. Heart drugs, women suffer from heart disease same as men. When you go to a cardiologist, he's not giving you one set of recommendations if you're a man and another if you're a woman. You have the same issues or the same biochemistry regardless of your gender.
Well,
And I mean,
have you heard about this thing called iron overload, where men get more heart attacks than women until menopause hits, and then women catch up, and the thinking is because women, whatever the technical term is bleed out the iron once a month, and so they don't get that accumulation of iron that will have cardiovascular effects. But then once they stop with menses, then all of a sudden they're accumulating more iron that has cardiovascular effects. Have you heard of that? Is that a fringe theory? All of supplements is a fringe theory, but
yeah,
have you heard
that could
about
be
that
true.
iron?
The other week I was looking at, because there was a talk on longevity at NBJ, um and most of the people on the panel were male. So I looked up to see, okay, what's the list of the longest living people in the world? Well, the top 50 of the people are women, or top 100 are
Right
women.
here. Yep.
And so you say, okay, well, I mean, how is that statistically possible that every single person that's the longest living is female? There's got
Because
to be a
we've been
reason
putting
behind
up with your bullshit for years.
it. That's right.
You would
That's
think
right.
that would kill you sooner.
Yeah. And so you
Makes
say,
us
okay,
more
well,
resilient.
maybe it's the fact that it's testosterone. There's a negative effect of testosterone, or maybe there's a protective effect of estrogen, or maybe
Maybe
it's,
men do stupider things that
yeah.
decrease their lifespan.
That's
Just
right.
just a thought.
Women are smarter,
That's right.
and they're more cognizant about health. But yeah, what's the statistical likelihood that the top 70 or 100 oldest living people are all women? No, there's got to be some underlying thing there. But you go to these longevity conferences, and everybody that's up there slapping their lips are men.
then I think we're gonna call out MBJ here in a very nice way and say, hey, next time you do something on longevity, give one of us a call so we can chime in too.
Yeah, it's inherent gender bias, like, and it's one example of kind of, a male dominated world, but what's the sense to it? Like, let's have some critical thinking around, like, maybe there's something about women that we need to be looking at that isn't just, what kind of sunglasses you're wearing, or red light in the morning, or whatever crazy thing they talk about these days.
And
Well, they
ladies and gentlemen out there in the podcast universe, this is why you need to follow Blake on LinkedIn for these hot takes that are really good.
they say that women make men live longer and age faster. So I don't know. Maybe there is something about longevity with women.
Yeah, there's women definitely have a protective effect on both of us, perhaps. So,
Yeah.
Maybe.
yeah.
So, if you, because I know we're kind of getting close on time here, if you were to give anyone advice on designing a clinical trial and what some of the most important factors to consider would be, what would you say?
Um, don't just dive into it with status quo. Talk to multiple people. Get quotes from different CROs for a similar concept. Because a lot of CROs, they're so focused on the study itself rather than whether the outcome is going to be helpful or uncover new answers, or answer difficult questions. And so that's your job as the sponsor, is to make sure that, well, number one, are they doing it right? Or because you spend all this time, you spend all this money, you want to get as much as you can out of it. So really looking closely, talking to different people, whether it's me or someone else that has been part of different studies, different types, is this, and then obviously, okay, are the outcomes, if the outcomes are positive, are they going to give us solid claims? And then are the claims they're going to give us, is it any different than what our 100 competitors are saying? Because ultimately you want to have something a little bit different than what everybody else is doing.
Yeah, I think you hit the nail on the head there, Blake, when you talked about claims. I think you should start with what's a great claim that we can make on the bottle or on our website. Like what is that claim? And then how do we design a trial that will elucidate that claim, right? Because that's where the beat meets the street, right? Is you want something that you could take to market. So
That's right. Product development, R&D, it should all start with the marketing people. And so hopefully your marketing people are smart. They're telling you what they think will sell, because we're not going to do all this effort for a product that's going to be discontinued next year. So the marketing people have to be smart, and they have to be driven, and they have to have the right answers as far as what they want to see in a product. And then everybody else, product development, R&D, we can follow that plan. We can deliver on that. But yeah, if you're gonna tell us, well, I don't know, here's our competitors, here's the claims they're making. You got to think a little bit harder than that.
cool. Think harder than that. I think we should end it with that. Blake Ebersole, he's going to make us think harder about life, the universe, and supplements therein from NaturPro Scientific. You could also follow him on LinkedIn for some really great conversations. I appreciate the heck out of you for that,
Absolutely.
Blake.
Thank you so much for joining us. I always love chatting with you.
Hey, it's been great, and can't imagine two better people or two better hosts. And yeah, this is what you're doing is great here, and
Thank
look
you.
forward to seeing more of it.
Well,
Cool.
we'll have you back again sometime.
Excellent.
Sounds good. All right. See you next time, everybody.
Bye.
Bye bye.
We are the Nutri